Bamboo vulgaris, also known as common bamboo, is a fast-growing, easy to genetically modify and highly practical and useful plant, which is specifically why it's key to our project. Today climate change is the greatest issue humanity is facing. We are constantly working against the clock trying to preserve the nature one can only find on this planet.
Greenhouse gases are necessary to keep the Earth's temperature above the freezing point. They do this by capturing the heat from the sun and preventing it from escaping back to space. However, over the years, the activity of humans has caused the natural levels of these gases to rise, causing an increase in the Earth's temperature, which we also know as global warming. The greenhouse gas, CO2, has been the main target as according to the statistics gathered by the European Parliament, CO2 was responsible for roughly 80% of emissions in the year 2021. This gas is released from the combustion of fossil fuels like petroleum, coal and crude oil. And ever since the onset of the Industrial Revolution in 1750, there has been a noticeable increase in the concentration of carbon dioxide in the Earth's atmosphere, surpassing 35 billion tonnes of CO2 released every year.
We propose a potential technique to elevate the efficiency of photosynthesis in bamboo grasses by replacing the native rubisco with rubisco from red algae such as G. munilis and G. silicornia that could potentially reduce the amount of these emissions in our atmosphere. The crucial but yet ineffective enzyme for fixing CO2 is frequently growth limiting within photosynthetic plants. Red-type rubiscos show advantageous energetic properties such as the best carbon fixing ability of any plant and high catalytic properties. For the editing of the bamboo genome, agrobacterium-mediated transformation will be used. The TI plasmid will be modified so that it includes the G. munilis genes, RBCL and RBCS, coding for the small and large subunits of the algae rubisco. Furthermore, the gene GMCBBX, which encodes rubisco activase subunit from G. silicornia, will also be incorporated into the plasmid. To completely inhibit the expression of the native bamboo rubisco, we will integrate short interfering RNA and its respective suppressive promoter to degrade the native rubisco's mRNA. We will add FL1F and FL1R primers with the restriction site MAN-B1. The recombinant plasmid will be reinserted into the bacteria and then, with the help of VIR genes, the tDNA will be cleaved from the plant cell. Following this, the plant cells will be cultured. The transformant will contain rubisco L8S8 assembly. To examine the efficiency of the small and large subunits transcription, we will use real-time PCR, Western blotting, and gel electrophoresis. Genetically transformed bamboo, just like that.
These trees do a great job at removing the carbon we relentlessly pump into our atmosphere and using it to grow and proliferate. However, they're struggling to keep up with us.
Our photosynthesizing organisms are some of the only things preventing our world from going from this to this.
Our modified bamboo products have a great ability to be commercialized. For example, bamboo can be used as a sustainable alternative for wood in construction.
It requires much less water and nutrients to grow than other natural materials, such as cotton or wood. It also has the ability to be reharvested each year without damage to soil microbiome or need for replanting.
But our genetically engineered bamboo will allow us to prevent this.
If we're able to implement it. Bamboo can also act as a biofuel, offering another solution to our climate crisis.
The materials we use in life, with proper investment, we can have a serious impact on our need for affordable clean energy, industry, innovation and infrastructure, sustainable cities and communities, responsible consumption and production, and perhaps most importantly, climate action. And not to mention, it's also delicious. That's right, our quest to help our planet doesn't just end there. We want to use our increased biomass to give back to the people. That's why we also want to contribute to attaining zero hunger with our nutrient-packed perennial. And so, we hope to secure your funding to allow us to work on combating climate change, one stem at a time. Choose right, choose Bambio.